And thank you so much for joining us tonight. My name is Kate Bruns, and on behalf of a lot of people here, Harvard Bookstore, the Harvard University Division of Science, the Cabot Science Library, and WBUR, I am very excited to welcome you to our program tonight with Donna Jackson Nakazawa discussing her latest book, The Angel and the Assassin, the tiny brain cell that changed the course of medicine, in conversation with Harvard Medical School's Dr. Beth Stevens, moderated by WBUR's Carey Goldberg. In just a moment, I'm going to turn things over to Carey, who will be introducing the program tonight. But first, I would just like to say a few words about this series and how you can learn more about our upcoming spring season. This Science Book Talk series features talks throughout the academic year by the authors of recently published science-related literature. Just this next month, we are very excited to welcome linguist Daniel Everett for his book, How Language Began, and renowned physicist Brian Greene for his new book, Until the End of Time. If you'd like to stay up to date on the series and our newly posted talks, I have a couple options for you. Nearly all of our events are filmed and recorded for the public. You can access them at the web address that I've written on the top here, science.fas.harvard.edu/booktalks. Or you can find announcements about our spring events lineup on harvard.com/events/science. And lastly, you can learn about the bookstore's other non-science book talks, which take place nearly every day, by visiting us at harvard.com and signing up for our weekly email newsletter. Tonight's talk is going to be followed by a Q&A, after which we'll have a book signing and refreshments in the library across the hall. And if you haven't picked one up already, we have copies of The Angel and the Assassin for sale in the back of the hall and in the library as well. And as always, just a couple thank yous. Thank you to our partners here at Harvard who make this series and the very important cheese trays at the reception possible. And thank you to all of you for being here, for purchasing books that support independent business, and for affirming that science does matter. Finally, just a quick reminder to please silence your cell phones before the talk begins. So now I'm excited to introduce Carey Goldberg, host of WBUR's CommonHealth section. Not only is Carey the co-author of her triple memoir, 3 Wishes, but she's also been the Boston bureau chief of the New York Times, a staff Moscow correspondent for the Los Angeles Times, and a health and science reporter for the Boston Globe. We're so happy to have her here tonight. Please join me in welcoming Carey Goldberg.
Thank you. Thanks, Kate. Thank you, Kate. Thank you, and speaking very unofficially for WBUR, I'm sorry about all the impeachment that we've been airing. I know it's too much. Now, before I even introduce our amazing guests, I'd like to get a little sense of the audience. So I will be offering you 3 choices. Number 1, I am already intimate with microglia. Number 2, I kind of vaguely know what microglia are because I've got a nice grounding in neuroscience. Number 3, what are microglia? Some kind of small pasta dish? Like, what is this? Okay, so number 1, I'm already intimate with microglia. Please raise your hand if you are. Oh, okay, so about 10. Number 2, Rob, yeah. Number 2, I know what they are, but not much more. Okay, so that's the sweet spot. Number 3, what the heck are microglia? Oh, that's a big one too. Okay, great. So then, great, that's a great starting point. I would just like to say, as I was thinking about this amazing book, there comes a time, I can remember about 5 years ago, I said to myself, okay, I do science reporting for a general audience. We have now reached the point where every well-informed person needs to know what CRISPR is. And that came to pass. And I would say we've reached that point with microglia. So it's a particular great privilege to be hosting this evening tonight. Our guests, on the right, Donna Jackson Nakazawa is an award-winning journalist, internationally recognized speaker whose work explores the intersection of neuroscience, immunology. So I was thinking also it combines my favorite organ with my favorite system, right? So it's the brain and the immune system. And also she writes about human emotion beautifully, I would add. She's the author of 6 books. Tonight's The Angel and the Assassin, the tiny brain cell that changed the course of medicine. Also Childhood Disrupted, how your biography becomes your biology and how you can heal. Also, The Last Best Cure, The Autoimmune Epidemic, and Does Anybody Else Look Like Me? A Parent's Guide to Raising Multicultural Children. And her writing has been published in every major publication. So please join me in welcoming Donna. And the blonde on the left is Dr. Beth Stevens. She received a BS from Northeastern, Ph.D. from University of Maryland, postdoctoral fellow at Stanford, currently an associate professor in the Department of Neurology at Harvard Medical School and a research associate at the Kirby Neurobiology Center at Boston Children's. She's also an institute member of the Broad, and her scientific papers have appeared in such journals as Neuron Science, Proceedings of the National Academy of Sciences, Nature Neuroscience, among others. So this is the biography that was given to me. I don't know how you can be so distinguished that you don't even include your MacArthur Genius Grant on your short biography, but it must be nice. What are you talking about, the angel and the assassin?
Well, I'm trying to put in my mind that the majority of you know what microglia are, so I think we have to start a little bit there. And microglia are these cells in the brain that were miscategorized for a long time, thought to be boring housekeepers, or as a science journalist would say, you know, kind of like the entourage that takes care of a movie star. They just took care of neurons and helped them be healthy. But then someone discovered that they actually had this dual capability. And they're capable of not only repairing and restoring neurons and carting away dead neurons, but also munching away and eating at synapses, the synapses that we need for clarity of mind and joy and cognition. And to me, that makes them, in their good state, in homeostasis, the angels of the mind. But they can also be the brain's untimely and sort of blind assassins. And actually, my editor from Random House is sitting right here. You knew I would do this to you. So one day, she emailed me or called me, and she goes, sorry, Marnie. I was in the shower, and I thought we should call this book The Angel and the Assassin. That's what I'm talking about.
Got it. Great. Makes sense. Beth, do you want to elaborate?
Yeah, sure. I think so. Put my neuroscientist hat on and thinking about like how I got interested in these cells, and what I knew when I was a graduate student was that these cells are our resident immune cells. So, our brain is made up of lots of cells, right? We have neurons, we have glial cells, the non-neuronal cells of the brain, and then we have about 10% of the cells of the brain are made up of these cells called microglia, which in many ways are very similar to cells in the immune system that help to protect our body against a bacterial infection or fight off infection, things like that. And it turns out that there's about 10% of our cells in the brain are these microglia. And much of what we knew about these cells, like back when I was a graduate school just starting to learn about the brain, was that they mostly got activated or disrupted in the context of an infection or disease. And we just didn't know a lot about what they normally did. And so I think now we appreciate that they have so many roles in the brain, and that not only do they play important roles in disease and injury, where they become really activated, but what we're starting to appreciate is because they enter the brain really early in development, in fact, they're the first glial or non-neuronal cell in our brains, like before birth, that tells us that these cells must be doing other things, and that's what got me interested in them. And what we and many others in the field have uncovered over the last decade in particular is that these cells play really important roles in building the brain and wiring the brain, and at the same time, they can also unwire the brain. And so I think that's the sort of new discoveries over the last many years, thanks to many in the field and new tools and new ways in which to study them, is starting to uncover what they normally do, which is a lot of things, which we can talk about. Mm-hmm. But then understanding what they normally do, I think, is really critical for understanding how they go awry in disease. And I think you need to have both pieces of information.
And so jumping to how they go awry in disease. So the book intersperses the science, including in Beth's lab and other labs, with patients whose problems, mostly psychiatric problems, actually, right, are linked to possibly microglia and dysfunction of microglia. So can you sort of paint that picture? And also, though, in particular, what do we really know about the extent to which these very complex phenomena are, in fact, linked to microglia dysfunction?
Great question. And so I covered patients, followed them for 1.5 years, 3 patients. And they were suffering from depression, panic disorder, anxiety disorders, cognitive issues, autoimmune issues. And really what I was trying to show was this link, which we do know, between chronic physical inflammation in the body, so diseases like MS and lupus, and that revved up inflammation, which we know gets our immune system and immune cells to possibly overreact when they're overtaxed. And these patients who had autoimmune conditions also had cognitive issues and psychiatric disorders. And I wanted to show that link, even though, to get to your question, the really screwdriver down proof of microglia, to the best of my knowledge, is in Alzheimer's and schizophrenia. So I think I have to give that over to Beth in terms of, there was no place in the book where I said microglia cause depression. Now I know there are genes that are expressed through microglia that are implicated in depression. But the way I look at it as a science journalist is epidemiological research, these people with this have this. Then you start to narrow it in, right? And get to who's looking at things on a disease mechanism. We're not there yet for all these diseases, but we are there for some of them. And I think that Beth can speak to that.
Yeah, I think so. So I think one of the things to say is, neuroinflammation is something that's very broad. It's relevant to a lot of diseases. Sometimes, in fact, in most cases, that might be secondary to an initial insult, right? But it doesn't mean that it's not important, because if neuroinflammation starts to go awry, when that process happens, then other issues can happen. And it's often could be as important as what started the whole thing. So on one hand, some disorders where the inflammation isn't the driver, it's still important to think about how to try to get that back into check. And it's been really hard to do that. And that's very broad, from a brain injury in terms of a traumatic brain injury to a neurodegenerative disease, right? And I think one of the things that's exciting now, and this is where some of our own work and others as well in the field, is that even in Alzheimer's disease, where this is one of those that we know from certain cases where it's the familial form, or where the very rare percentage of people have these genes like presenilin, that was largely thought that this microglial part was sort of secondary. But what's now happening, thanks to a lot of new genetic studies from patients over the last many years, where geneticists are able to look to see what are the genes that are causing most of Alzheimer's, which is like late onset Alzheimer's. In this particular case, many of those genes are either expressed in or exclusively expressed in or enriched in myeloid cells or microglia. Now that's a good example of a disease where now even those who've been studying Alzheimer's for their whole career are starting to take notice of microglia, because they're like, wait a minute, this can't just be secondary, because the genes that cause late onset Alzheimer's are expressed in these cells. So that's a good example where there is some evidence to suggest that they may indeed be driving some aspects of Alzheimer's. It's less clear in many of these other neuropsychiatric conditions, because the genetics aren't squarely pointing to them, but it doesn't mean that they're not responding and causing problems. And I think that's part of the thing we need to be thinking about is, are they initiating or driving, or are they sort of getting revved up? And in any case, whether it's one or the other, we still need to figure out ways to know the answer, and to also start to think about how to control them and get them back into their good roles in the brain. And I think that's one of the, there are some disorders that it's more challenging to do, but we can learn a lot from the diseases in which we know more about their role, and then ask whether understanding that could be applied to the more complex diseases, like, say, schizophrenia or neuropsychiatric disorders.
Can I just say one thing that in the reporting that I just want to clarify? Every time a researcher like Beth or Yoni Kipnis or anyone would say to me the word neuroinflammation, I'm a science journalist, it confused me for a little while, because we're taught red hot, painful, swollen, right? So you're hanging a picture and you hit your thumb and it gets red hot, painful, and swollen, and you're thinking that's neuroinflammation. The brain, neuroinflammation, is a little different. And in the role of microglia in neuroinflammation, they're spitting out these neurotoxins and eating away at wiring at synaptic connections. So I just wanted to say it's a little different.
Yeah.
And when that, as best I understand it, when that inflammation or that microglia engage in runaway inflammation, that's that either primary or secondary process that we think is involved in all of these diseases.
And even inflammation can be both good and bad.
Right.
So in other words, these cells release a lot of these factors that can cause sort of the bad kind of inflammation, but they can also, in certain contexts, release things that can help. So that's part of the process of trying to understand that word is really loaded, I guess is what I'm trying to say.
Neuroinflammation.
Neuroinflammation. And so literally, that doesn't necessarily tell you what it's doing. And so that's part of trying to parse that out. But we can say that these cells are definitely one of the mediators of inflammation, both the good and the bad, because they actually produce a lot of the signals that lead to that in the brain.
So if microglia and neuroinflammation more generally were involved in the genesis of Alzheimer's, how would they be doing that?
So
even just speculatively.
Yeah, so I can tell you, what we sort of know at this point. There's already been evidence of microglia's sort of beneficial or good roles in the context of Alzheimer's. Like, for example, there are amyloid plaques, toxic proteins that build up in the brains of those that have Alzheimer's disease. And one of the things we know that microglia are important in doing is to eat or clear away that plaque. So you'd really want your microglia, when they start to encounter a toxic protein like amyloid, to kick into action and be like, I'm going to get rid of this. And so them clearing and getting rid of it would be something you'd want your microglia to do. What we're finding from both animal model studies and other lines of evidence is that in the context of Alzheimer's disease, microglia, in this case, are not responding and clearing those plaques the way they should be. So in this case, you want to find a way to get them back over clearing, getting them back into that clearing mode, right? But at the same time, as Donna pointed out, them eating plaques, right? You don't want to get them so revved up that they start eating things they're not supposed to, like synapses and healthy neurons. So how do you deal with this yin-yang, this balance? And I think that's been the challenge. And so, I think in the case of Alzheimer's, where we actually have some evidence of like some of those genes now, like we know genes, we know they're expressed in these cells, so scientists can start to say, well, let's start to tease this out and ask, what is this gene doing to the cell? Are they doing good or bad? And then start to parse that out. This approach is really powerful because if you get enough scientists working on this really systematically, we can start to say, our goal would be, let's get them to react, clearing plaques, but not overreact. And that's part of, I think, it's a hard problem, but it's one we have some traction on now.
Donna, do you want to add something?
No, I think that's the, as a journalist, your job is to start a conversation. Does that make sense? You do that too.
Yes.
And you're not setting out to have all the answers. You're starting a conversation so that there are news stories and meetings like this and people asking you questions, so that collaboration and that funding and that just new way of looking at possibilities of healing comes not in that 20-year gap that scientific philosophers say happens between the lab and the clinic, but like if I could just help Beth and her colleagues shorten it up to 4 or 5 years, I think that would be amazing for the patients who are running out of tools and hope.
Yeah. So from Alzheimer's to schizophrenia, if Alzheimer's might be microglia not clearing enough, schizophrenia, it sounds like the story is more over pruning. Can you tell that?
Yeah, that's again a hypothesis. And in that case, it's another example where human genetics has, through a completely unbiased way, have shown a role of some of these immune molecules. And some of them are linked to microglia, not directly, but indirectly, in that they are responding to some of those immune molecules in the brain. So that would be an example, and I think in Alzheimer's, I should say, even though we just talked about plaques and clearing, one of the earliest hallmarks of Alzheimer's and many neurodegenerative diseases, meaning long before you even see the clinical cognitive symptoms and a lot of those plaques building up in the brain, is that you start to lose synapses, the connections between neurons, right? And so this seems to be a common hallmark across many disorders, ranging from schizophrenia, in the case of an overpruning idea, to Alzheimer's disease. And so in this case, this is an example where losing your synapses, which is the strongest correlate of cognitive decline and memory loss, for example, this is something that, to my knowledge, there's no drug targeting that part of it. This is a very early event in the context of Alzheimer's disease. And it also is hypothesized to play a role in neuropsychiatric and neurodevelopmental disorders. So understanding microglia's role in synapse loss, it could be another way of sort of getting at this early event that's happening. So in other words, there may be different mechanisms regulating the synapse eating from the plaque eating, and I suspect there probably are. And that is, if we could figure out the difference, then we could target those two things differently. And I think the take-home would be, microglia are not all created equal, and if you just say, I just want to get rid of them, or I just want to do one thing to all of them, that might not be the solution. But if we can understand the different flavors of microglia, the different states they have in the brain at different times, that might then give us a more targeted approach. The precision medicine idea, where you just want to go after the part that's bad, target that, but leave alone all the good functions that they have in the brain, or try to promote the good functions they have in the brain. Mm-hmm. And I think that's sort of, in a way, one way to sort of tease out the sort of plaque from the synapse in the case of Alzheimer's as well.
And if we could do that, like if we got much better at targeting microglia in ways that could be beneficial,
yeah.
Donna, what do you think, what is the potential impact on especially neuropsychiatric disease?
Well, I think that so big picture. So the way I look at it, Beth is tiny picture, and she takes it to the big picture. I'm coming from the point of view of patients and people who are suffering and their families who are suffering. I think if we could beam forward to a time that I imagine, if I can do that, where this science is more realized. Mm-hmm. And I would talk to Beth in her office and hear wild things like, very exciting, like at Broad, they're working on the possibility of looking at what I think of as barcodes. Remember, I'm a writer, I use analogy, on microglia cells as they begin to change states. And that's not here right now, but it's in the works. And if we can get to a place like that, just imagine that we have a family with bipolar or schizophrenia or intractable depression, and the grandmother in that family has Alzheimer's, and a 12-year-old is coming in, and we have the ability to look in the future, this is not here, at microglia changing states. And we know that the brain is really our seventh sense. It's reacting to the world around us in the same way that our immune system is. The role of microglia in this reactivity or this brain immune response, we may not be able to tie it all down, but imagine a time when we can tie it down better. And a 12-year-old goes in and has a blood test the same way they have a cholesterol test today, or a white blood cell count. And I am simplifying, I know that, but it's big picture. And a pediatrician goes, well, there's a lot of microglial overactivity. Could be good, could be there's an infection, could be bad. Maybe we'll have those brain scans you and I talked about, where we'll be able to look at synaptic connectivity and we'll go, hey, that's not looking so good. Let's be prophylactic here. Because as Beth just said, we see this synapse loss years before symptoms. And I think the promise of this for families and patients is to get in before some of these diseases happen, in addition to treating them better when they do happen. Those are both really important and exciting.
So, sorry, please forgive me, I have to ask the goop question, though, which is, I mean, this is all amazing work, but it's, for clinical application, it's very far away.
Very theoretical.
Yeah. So If what you're saying is also that it seems fairly clear that neuroinflammation is important in a lot of these diseases, is there anything that can be done now to affect neuroinflammation?
Well, in the reporting that I did, I followed lots of patients trying lots of things.
Yeah.
And that gets back
So
to
tell
the
about that.
epidemiological, right?
Right.
So again, we're not now in the lab looking through high-resolution microscopes. We're looking epidemiologically, and then we're making inferences. Some of the patients I followed were doing transcranial magnetic stimulation, which reboots neurons, and we think theoretically, or at least some of the people I talked to thought theoretically, microglia would have to be involved in this rebooting. Now, theoretically, if patients all of a sudden who have had intractable depression, have tried 30 medications, haven't been able to get out of bed, can't have a doorbell ringing in their home because it's too overwhelming for them, can't eat in a restaurant, and they go through a treatment and their lives change, then hypothetically, we would think that cells in the brain are behaving better. Other things, are very hypothetical, like gamma light flicker therapy. I've got to tell you, who's read the book? Anybody? Okay, number one question I get from people is, when can I get gamma light flicker therapy?
They're doing it at the pick hour, right?
At the pick hour,
But
yes.
yeah.
And it is in clinical trials.
Can you do it yourself somehow? Like set it up.
Other things are available now, and I'm not suggesting anything, right? This is all theoretical. It all has to be done with a doctor, a psychiatrist, a team. But the idea that we can reboot our microbiome a little bit with intermittent fasting, safe fasting, done with a medical practitioner. There are now, Mark Mattson at Hopkins is tying it to the activity of microglia. Again, we're not screwdrivering it down on a under the microscope level, but I think opening up that idea, the bigger idea, that our brain has an immune response. We all know, right? We care for our body's immune system. In a little bit, well, I can say, I wrote a book called The Autoimmune Epidemic, okay? People did not think that our immune response mattered very much in terms of developing autoimmune diseases. Obviously, the immune system went on overdrive in autoimmune diseases, but it was thought to be impossible that that was affected by what we ate, or toxins in the environment, or environmental stressors, or chronic childhood stress. And now that understanding is really here, that our immune system is in this intricate dance every minute with the world around us. And our brain is too. And I think that's really the message.
But so you come up with a sort of what you call a unifying theory, right? And what it does is connect the dots between the immune system issues that are so common now and the brain issues that are so common now.
Yes.
But it's just a theory, right?
Yes.
At this point,
That's
like
why
it's a
it
lot of
says
correlation.
theory.
Yeah, it's there,
That's
right?
why
So
it
you
says
can't
theory.
really, yeah.
Absolutely. It's
I mean,
a theory, and just like any unifying theory. But if you talk to 20 of the top researchers right now in neuroscience, they're all talking about this cell. They're all talking about this cell. And as a reporter,
very hot.
you listen to that. Yeah.
Do you see that, Beth, also? Like it's just sizzling?
Yes.
It's sizzling.
So I guess two things I would say. I think there's been more and more, a lot of actual scientific evidence that there's this interaction between the nervous system and the immune system, and it's influencing brain function. And of course, a lot of that's being done on mice and animal models. We can't get into people's brains and be able to do this over time the way we can with models. But nonetheless, I think what has really changed the game is, again, I keep going back to genetics. Why does that matter? When genetics is in an unbiased way saying that the causal genes or genes involved in risk of a disease are expressed exclusively in this cell type, and Alzheimer's, that's a couple examples of that, then that's telling us it's not just sort of, it's real. Now the question is how? How is that working? And so that's where I'd say we're a little bit far from like taking that information and making a medicine, but there's a lot of interest and a lot of work in many labs across the world working on this. And that's in part because of the genetics nominating this, and it's also because more and more evidence is mounting that they are critically important in some of these diseases. So that I feel strongly that there's evidence for that. That's not just a theory. The theory is how you take that information and like, what does that really mean for sort of something like complex neuropsychiatric? Now that's really hard, right? But the one thing I'll say, and I guess this is an optimistic way of thinking about what could we do now versus later. There's a lot of work that needs to be done to try to move forward all of the data that's emerging from many, many labs to try to understand how to target these cells in that very specific way. That we really are working towards that, and I think we are making some headway. But in the short term, what I do think could come from a lot of the work that's already been done is a way, as Donna was just pointing out, what if we had a way in people to non-invasively being able to read out microglia changes in the brain.
Yes.
In fact, this is relevant because no matter whether you believe that they cause disease or they respond to disease, I think we can all agree they change in disease. There's a lot of evidence for this. But the problem with people is we can't right now put a person into a scanner and say, what is your microglia doing? I can do that in a mouse very nicely. I can do that in other model systems, but in some cases, it would be very beneficial to know. I'll even take an extreme example where you have a traumatic brain injury, right? You have an insult to your brain. We know the microglia become activated. Wouldn't it be great if we could track that in patients with an imaging dye, right? A PET ligand or a brain imaging tool that can non-invasively put someone in a scanner and watch microglia, see where they are in the brain, where they're activated, and then a week later or 2 weeks later or a month later, put them in again and say, well, did that inflammation of those microglia, are they different? You could say the same for Alzheimer's, where we know they're important. Wouldn't we like to know if a drug in a clinical trial is impacting them in some way? So what I think isn't so far away is that we could generate and develop new tools, like new imaging tools, to be able to track and monitor microglia in a non-invasive way through these brain imaging sort of tools, much the way you can with amyloid and Alzheimer's disease. And how do you get those new tools is you need to do all this research to try to identify what those markers might be, and then to try to translate that into making a new PET ligand, for example. So there's a lot of work on that happening now, so I don't think that's that far away. Or if you knew what to read out in blood or cerebral spinal fluid, where those materials are available, you would know what to monitor as a biomarker. That would also be really helpful. So the biomarkers are really key, right? Because then you can actually read things out over time. You can look at progressive changes over time, or you can look, in some cases, diagnostically. And that, I think, isn't so far away, given all the work that's happening in the field.
And I want to go back to your question, because wouldn't that be amazing, even if we couldn't show exactly what's working in what patient, because we can't go in their brain and look at their neurons and microglia, we just can't do that. But what if we could use some of these tools and we could say, wow, we have patient A who's showing early signs of Alzheimer's, and we threw this at her and this at her and this at her, and she did these different things. And this is working. She's getting better. Or what if we saw somebody wasn't improving and we tried something else? And using these tools, we could see that that was working. Because one thing you learn after reporting on patients for 30 years is the same thing doesn't work for everybody.
Yeah.
Different things work for different people who have the same symptoms and the same disease. And a tool like that, even if we never tie every single thing down to looking in someone's brain on a second-by-second level at these trillions of cells and synapses, if we can tie down that we've changed the activity of these cells in a way that's correlated with symptoms improvement, that's a game changer.
Yeah, I think that's true.
And that's closer.
Yeah,
Close.
So there are some PET ligands or brain imaging tools where people have said it's a microglia sort of PET ligand. but it's not specific for these cells. It's more of a kind of general readout for maybe neuroinflammation, but even that's a bit wishy-washy. So there are such tools out there now that are also being used in the clinic, but it's not specific for microglia only, and it's not really telling us what they're doing, and we don't know what it's reading out, so it's sort of a proxy. So imagine if we can get something more specific. So I don't think we're that far away from something like that, and I know there's many people working on it. But then if we had such a tool, then we could start to think about stratifying patients and starting to look in the right populations. Again, adding another biomarker with the existing ones in certain disorders would enable you to get more precise. And I think that is something that is much needed across many disorders and diseases.
Interesting.
Yeah.
So I have one more question, and then we will move to audience questions. So if you want to start thinking of yours, now is the time. So because a lot of what we do as journalists is simplify, and sometimes very violently, I'm feeling frustrated because it's not very simple. Like it's like the microglia can be acting in a beneficial way.
Imagine reporting on it for 2
I know,
years.
poor you. But it's like so. I guess my question is, so that biomarkers are important, ligands are important, but what might be the first instance in which there's a clinical application for our knowledge, for our rising knowledge about microglia that could, in fact, be medically helpful?
Well, I would go back to, I don't want to use the same story I used before, but I really think that if we begin to see neuropsychiatric changes in a patient, we can begin to ask questions about their step back from microglia. We can ask questions about their brain's immune health. And that is not a concept that has been out there in the psychiatric community, period. So we have not been asking these questions about the brain's immune health. We have not been adding that into neuropsychiatry. A patient goes in and they have very important medications that may or may not work, have a 33% efficacy rate, and the efficacy can go down over time, and they don't work for everybody. We have wonderful different forms of psychotherapy and CBT and DBT and ACE, all of it. And it is helpful, but a lot of people have intractable disease. And if we have missed this idea for the last century that the brain is an immune organ and it is our physical health, I think that opens up a different discussion in the psychiatrist's office, in the doctor's office, and it also catalyzes patients to ask their doctors about this. And
But what can their doctors do?
Well, I don't know. How are you eating? Let's look at your microbiome. Are you exercising? I mean, we're tying all these things down to the brain's physical health. And you have to remember, 10 years ago, the idea that the brain was immune privileged was still, that was dogma in medicine. So if we step back from this tiny little cell and go, wow, the research that Beth has done and others are doing in the field, and she's one of those great researchers who, whenever I say, oh, whatever Beth is doing, and she goes, and everybody else.
Yeah.
If we step back, it gives us a new paradigm for thinking, okay, that dogma that the brain is immune privileged, not ruled by the immune system, was wrong. That cannot help but change how we see these diseases.
Beth, I want to make sure you have a chance to give a pitch for data sharing before we get to questions.
Yeah.
Absolutely.
Yeah, so maybe talk about research instead of clinical.
Yeah, sure.
Yeah.
Yeah, I would say my one point to follow up with Donna, though, I would say that when you think about, if you want to target these cells, or if you want to start to think about how you could have an impact, I guess I would probably err on picking a disorder or disease where it's more tractable, where you sort of know either, these patients definitely have this, or there's some way you could read this out in a way that you could have an impact, even in a rare situation, that if you understand that really well, it could have broad implications for something really complex like neuropsychiatric. I think neuropsychiatric disorders are really hard. Lots of genes, the environment, gene combination, it's very hard to deal with. But imagine if we figured out how to either manipulate some aspect of microglia, whether that be neuroinflammation or synapse eating or clearance of something then that, if we could figure that out, it might then have broad implications in the harder disorders. So I do think the strategy of going after something where we know for sure microglia are definitely playing a key role, then figuring out how to have an impact, then we can start to say, with the biomarkers, how that goes to the more complex disorders. I just think the neuropsychiatric is really a hard one because it's so heterogeneous and it's even more complex than some of these other diseases.
So what would that thing be where we know the microglia are playing a role?
Well, I think Alzheimer's is an example of that, although that's a hard one too, but there's a lot of excitement, and this gets me to the bridge to the data sharing comment. Okay, so if you had gone to an Alzheimer's meeting 10 years ago, which is when I started sort of entering the field and started thinking about Alzheimer's disease, I would have been probably one of very few people in the room that were thinking about microglia and Alzheimer's disease, in part because it was too early and the data didn't really point to them yet. But what's really been remarkable over the last few years is how many people are, because of the data, are like, okay, let's get into this, let's try to figure this out. So how do we take a field that's been very dominated by these models or these hypotheses and start to say, well, okay, it's not just this, it's also this. So it's been really interesting to see how that works. And the other thing is because it's immune, you can't just do this as you're a neuroscientist because neuroscientists don't have a lot of training in the immune system. So how do you really tackle this? The way to do it is to bring together new communities to a field that's been pretty much focused on this and say, let's start mixing in immunologists and geneticists and computational people and people that see things from different perspectives. And that's how you tackle really hard and different kinds of problems. And that's actually what's been happening over the last 5 years in this really exciting way. But if you really want to make an impact, then you start to have to realize you can't work in silos. Like you can't have lab X or this university doing it and not sharing information. And you can share information by publishing a paper, but sometimes that takes a long time. So what we've really been trying to do, not it's just at the Broad, but in many different contexts, is bring people around the table that normally wouldn't talk to each other and say, let's really work together on this really hard problem. And it's already amazing what's been happening. And we're starting to share data, share information, share tools in ways that are catalyzing all this research to happen. And that's also, I think, critical if we're going to ever move forward in a rapid way. So I am super optimistic and hopeful because I think people are realizing that's the only way to really do this. And I think the data sharing aspect is key.
It's happening.
It is happening.
Nice to hear.
It's really happening.
Good.
Yeah.
Okay, questions? Marty's, yeah, one right here. Marty, got I would, before we start, I'm abandoning all dignity and just begging you to make these short and also to make them questions rather than statements. Yeah.
Experience moderator.
Yeah, I don't think. Don't make me be mean.
Short question. Getting information out of the brain, as you pointed out, is very challenging clinically. So is there any hope that there'll be something in the peripheral nervous system that'll be a stalking horse for the information you'd get from the microglia in the brain?
So the question is there any hope that something in the periphery can be helpful? Yeah, actually, yes, I do. And actually, that brings up another, again, kind of an optimistic thinking about how to do this, is because of some shared mechanisms from the periphery and the brain, even though they're different, there's definitely going to be unique things that are unique to the brain versus something that's happening in another organ or in the peripheral blood. We could imagine almost thinking about repurposing some of these things that have been happening in peripheral inflammation and then asking if we could test that in the context of brain and then develop ways to get those into the brain. I think that is actually already happening. And that would also be a way of fast forwarding so that you're actually repurposing things that could be already used in this context. Yeah, it's a great question.
One right here.
Okay.
Yeah, this question's more for Dr. Stevens. I think it's a really great theory about microglia and the overactive microglia in the formation of schizophrenia. But how does that tie in with medicines for schizophrenia, which is more neurochemical and works on dopamine? How do they link up?
Yeah, that's a great question. I mean, that's, I guess, getting back to the big challenge of schizophrenia and neuropsychiatric disorders is that there hasn't been any drugs yet that are targeting some of the cognitive impairment in schizophrenia, right? And a lot of this has been more in the context of neurotransmitters and signals between neurons. And the genetics, to be fair, has mostly pointed towards neurons and synapses, not microglia directly. So I don't think yet we can say how targeting the microglia is going to suddenly be a new treatment for schizophrenia. I think it's way too early for that. But that said, because at least some of the evidence from genetics of schizophrenia also implicates some of these immune pathways that also could be affecting microglia, there is a potential that if understanding how to target microglia could indirectly impact schizophrenia, I think that's one way to think about it. But I wouldn't start there. That is not the disorder I would go after, because the genetics aren't, like Alzheimer's, squarely pointing to microglia and schizophrenia. So I think where the genetics are, if schizophrenia is pointing to, however, are those connections between neurons, those synapses. And so how to start to think about how to target those, that's where I think a lot of people in the field and the Stanley Center and other places are starting to work on. But again, it's a harder problem because we can't really model that in animals. It's a very complex cognitive disorder that isn't easily modeled. And I think that's another big challenge with neuropsychiatric disorders. But again, that's where the biomarkers can be really powerful.
We have a question over here.
Oh, sorry, go ahead. Yeah.
I'm Jackie Olds, a psychiatrist, and I have been reading lately some of these books called things like Eat to Beat Disease, and they talk about the immune calming additives, mushrooms of various sorts, and they talk about the immune boosting additives or foods. And I just wondered whether you've tried any of those in any of your animal studies.
Oh,
do any of
days.
Oh,
yeah.
We were psilocybin. No.
We were talking
The
about the
anti-inflammatory diets are pretty hot.
Yeah.
No, go ahead, Donna.
Yeah, so I did not look at mushrooms or special foods. I think that, to go back to something Beth has pointed out, neuropsychiatric diseases are very complicated, and I think that everything that a patient can do with any disease, right, is helpful. And again, I've reported on patients with serious illnesses for 30 years. And when patients find answers, it's generally lots of things, right? Like, becoming ill is multifactorial: genetics, environment, stressors, trauma, diet, microbiome, toxins, infections. We can name dozens of things that play into disease manifesting in any individual. And usually healing is like that too. It usually doesn't come down to just broccoli. But that doesn't mean broccoli is bad, right?
I'd like to mention that you recovered from Guillain-Barré syndrome twice.
I did.
Right, which is amazing.
Yeah, and that was bringing everything to the game. And I mean everything, from a guy slinging me, putting me in a sling, and taking me and forcing me to do exercises that literally I couldn't do. My limbs would just rubber out. Gumby. My husband used to call me Gumby. But lovingly. I brought everything to that game. Everything. And I had a lot of cognitive changes during that time, which is sort of what clued me into this link between chronic inflammation and changes in the brain. I mean, my body wasn't mine, but my brain wasn't mine either. And at that time, the literature was saying that wasn't possible. But that's another story. But I think that you bring everything to that game. And I think that that's missing in psychiatry, I'll be frank. I think that's missing. I think bringing everything to the game is missing. So I wrote a book.
Yeah, go ahead.
So in the attempt to explain how these cells can either be very, very helpful or very, very destructive, is it useful to identify other systems or phenomena in the body or brain that can either be very good or very bad? For example, one thing you keep alluding to is the immune system, and it's good if it's at this level and a problem if it's overactive or underactive. But that idea that something is good if it's at one level and then destructive if it's over or under might just be one way of accounting for this very, very extreme difference between the building of the brain that you described in infancy and this kind of destruction of the brain in these other situations. So are there other parallel systems that are useful in thinking about?
Yeah, I think that this is true in many cases where I remember another question that you raised. These microglia in your brain, although there are some differences in those cells in your brain than the ones that are circulating in your blood, for example. So because their environment is different, like the peripheral nervous system or the periphery is different than your brain. However, there's some conserved things you can learn by studying them in other tissues. So I think there are ways to be able to use that to be able to start to model this. But the other thing is, I think also identifying which microglia. So in other words, there's populations of the microglia that are surrounding those plaques that we might be able to target those specifically, and that's another way of getting at it. So it's not all microglia. And I think that's also something that we can model in the laboratory to try to understand. And I didn't get into this because it's more kind of getting into the details. But just as an example, we can now take blood cells, right, from blood, and there's actually a kind of a niche or a stem cell that comes from blood, and you can actually grow it and actually make it, create it into a cell that can be converted into different brain cells. So we can actually make a microglia-like cell in a dish. Now, of course, this is very different than the one that's in your brain with all the other cells, but if it's from the same patient that has a disorder and we can get the blood from that patient, we can actually try to understand what's different about that microglia, that person's microglia, from someone who doesn't have the disorder. And that's a great example where you can start to model mechanism in a very systematic way to try to understand what it is that's different about it, very systematically. And then you come up with hypotheses that then can be tested in different ways. And again, we didn't have the ability until very recently to do that. But now we have the technology and the approaches where you can start to use that as one way to try to understand what's going on with that person's microglia or myeloid cells. So I think that's one example where you can start to ask, well, if we push it this way or that way, what drugs are going to push it which direction? And we've developed ways to be able to study that in the laboratory. That's actually pretty relevant to the human because it's their cells, right?
Kate,
Is it my turn?
Yeah.
So could you explain any connection that you know of between transcranial stimulation and traumatic brain injury?
I actually can't do that. I can't. I actually don't know the data on TMS and brain injury. I think TMS is fascinating, and I think it's been really helpful in depression. And I sat there and was a fly on the wall for a patient who went through 30 treatments. And it was mind-blowing to see how it helped her. And I'd watch the real movie of her brain coming back online in areas of the brain that were not connected.
Right.
It was wild. But I talked to Alvaro Pascual-Leone, who did a lot of the research here.
Here at Beth Israel Deaconess.
Yes, who really started the field of TMS. And he cautioned that, and I put this in the book, that in bipolar, we maybe don't want to do that, right? We don't know enough. It goes back to what Beth said, we don't know enough. And so I wouldn't begin to comment on traumatic brain injury. I just wouldn't do it. I don't know enough.
You're not aware of
I'm
studies
not
looking
aware
at
of those studies, so I can definitely do what Carey and I do and get on the National Library of Medicine and find out what's out there. But I don't know, so I wouldn't want to comment.
Yeah, go ahead.
I just wanted to go back to identifying specific microglia. And since it is an immune response, I was curious if in research it points to perhaps a virus or a viral element that it's fighting, and that it could one day lead to something like a vaccine for Alzheimer's.
Yeah, there is a sort of an infection theory now from some groups that suggests that there may be a pathogen that kind of stimulates this whole plaque and process. And again, this is all work still in progress, and I don't think that's going to explain all of Alzheimer's disease, but could there be some cases where this might be happening? Possibly. But I guess I also wanted to say one thing that just got sort of triggered by a couple of questions, is that when we talk about, again, it's the way you define questions, like the immune system in the brain. I just wanted to say, and I should have said this from the beginning, that even though traditionally we think when we talk about immune molecules or immune cells in the brain that it must be something coming from the immune system into the brain. Actually, that's the part that got me interested years ago, is because in the brain, without any disease, without any injury, no breach between the barrier between the two, there are a whole host of immune molecules and immune cells, microglia, that normally live and exist in the brain. And I think that's what's really changed the way scientists, neuroscientists like me, started thinking about neuroimmunology. And that is that I don't even know what came first. We called a lot of these molecules immune because immunologists studied them and discovered them. But if you go back now and look in the brain, so many of them are expressed by nerve cells, neurons, and glial cells normally. So which did come first? I don't know. We probably can go back and do some evolutionary tracking to figure out which came first. But the point is that just like in the body, the immune system's meant to defend and to respond, all of those molecules and cells in the brain are also meant and built to do that, to protect us or to respond to changes. And I think that's one of their good roles. That's right. And so under normal conditions, all kinds of changes happen in the brain, and you'd want the system to be working this way. The difference is when things go awry, either through genetic reasons or through an environmental reason, then what happens is sometimes that can go out of check. And so instead of just like responding and then coming back, under certain conditions and in some people, it can go out of control. And I think that's kind of what we're talking about. Mm-hmm. And it doesn't necessarily mean you suddenly had a huge infection or suddenly your brain barrier just opened up and things came in. That can happen. But in a lot of these cases, that's not what we're talking about. We're talking about the brain's own natural immune system that normally does all kinds of really important things, but sometimes those things go awry. And I think that's the new realization and almost like a change in dogma when you start using words like neuroimmunology. It doesn't mean what it used to mean. And that's been exciting, but it also means, wow, we need to learn a lot of things.
And a lot we don't know.
A lot we need to learn.
A lot we don't know.
It's so weird we ever even thought the brain was immune privileged.
Exactly. Like that's like,
why would that make sense?
I found a paper written in 2008. It said, it really just said, it's categorically immune privileged. 2008,
Yeah.
You know, it's in science, that's the blink of an eye.
it's still in textbooks, by the way.
Yeah, well.
Is there any evidence that microglia also go after the other toxic protein that builds up in the brain in people with Alzheimer's, the tau tangles?
There's a lot of interest and work being done to ask that question. The amyloid there's more evidence for, but tau similarly is another one of those hallmark proteins that can not only build up, but it can cause synapse loss and a lot of problems. So the question that many researchers are starting to ask, including my own lab, is how microglia are handling the tau. What do they do when there's tau building up in the neuron? And there's also tau that can come out of the cell. How does that change microglia? And again, using model systems, we can systematically start to ask that question. And we can start to block these pathways in microglia and see how that changes tau pathology or tau function. So again, it's a little early compared to amyloid, but a lot of work's being done on that very question. And you could say the same thing for Parkinson's and synuclein. There's a lot of toxic proteins and different disorders that can build up. And microglia definitely are responding to it. The big question is how, and how do they do that? And so I think, a lot of work's still being done, so I can't answer the question specifically, but I think it's a really important one.
If toxic proteins build up, does that mean that the microglia must be malfunctioning?
Not necessarily, but I think if a toxic protein is building up, I'd be very surprised, especially if it was locally around where it's happening, that they wouldn't be responding to it, right? And so the question is, what do they do with it? And maybe when there's too much of it, or if they're not able to continue to help, then the system kind of shuts down.
I think we have time
for a couple more.
One last question.
Oh, one last one? There's one down here, yeah.
I thought Riley was going to ask a question.
Work done on even mild concussions? Are people looking at the involvement with microglia? Or?
Yeah, at STAR, Research Center at the University of Maryland. Alan Faden is working expressly on concussion, mild concussion, and moderate concussion. And Alan has done a lot of work, Alan Faden, in looking at the response of microglia and trying to, in some of the similar ways to what Beth is talking about, trying to gauge how overactive microglia are in response to what might seem like a mild concussion. But if we can look at how active microglia are and take some kind of a, big picture shot at their overactivity, he's theorizing, that this certain levels of overactivity are saying, hey, this is a bigger deal than we thought it was, right? Like it could be you're hanging ornaments or new drapes and fall, and it looks like a little bump. But with some similar tools to what Beth was talking about, looking at factors of microglia that, as I understand it, I always like to say as a journalist, don't you? As I
barely understand it usually.
As I barely understand it, descend into the bloodstream and can be measured. Don't run to your doctor to have this done because it's not there yet. But that's what he's working on. And his hope is similar to what Beth is talking about with Alzheimer's and what we hope one day with neuropsychiatric, even as complicated as it is. He's working on that with concussion so that he could say, this is more serious than we thought. And also come back, retest, and go, okay, our concussion protocol here is working or not working. That's a game changer, right? Because we don't know right now. If we send a kid who has an injury from a soccer ball back on the field a week later because he looks like he feels better and he's not having headaches and he doesn't feel dizzy, we really don't know. And if he has a secondary concussion, then we're talking about, as I understand it, runaway inflammation, which may be microglia-led neuroinflammation. Even if it isn't microglia-led, microglia are going to get in on the party and cause havoc. They are going to mess up the house. So being able to know how much they're messing up the house is going to be a game changer. Really interesting research. And he's looking at anti-inflammatories, which might be able to be introduced immediately after concussion in emergency rooms to help calm down neuroinflammation.
Do they cross the blood-brain barrier?
He thinks that they do. So it's early days.
I think if we were ever going to pick, I think if you're ever going to, a great example of where that imaging biomarker I was just mentioning, where you know something happened, there's symptoms, and then you want to see something non-invasive that relates to a cell that you'd want to be able to ask how, because you could localize it even, and then you want to know a month from now or a year from now or 5 years from now, what do they look like now? Like, I actually think that is a really great example where if you had a non-invasive tool to monitor these cells, that would be really, I think it would be game-changing. And then one could say the same for other disorders, but the concussion, where there's clearly evidence of multiple concussions lead to cognitive problems, the problem has always been we can't know what part of the brain, what's really happening in there, because you can't go in, right? So I think that's a good application of if we could get a good imaging biomarker, that could be super helpful.
Okay, we need to wrap up. I'm going to give you each a final challenge. You each get one final sentence.
Oh boy.
That is the answer to this question, why do microglia matter? Oh. Donna, you should start because you've been doing a lot of wordsmithing.
I think they matter because for me, from my perspective, I tell stories. And when you tell stories, you need a main character to help. No, I mean it. Yeah. You need a main character to follow. And I can blame Marnie again because when we were struggling to figure out how to tell this story about this shift in dogma about the brain, that's where it started. I started with brain flame. I was like, we didn't think this was possible, that neuroinflammation was possible. We didn't think the brain was this responsive immune organ in this dance with the environment. And then she was like, well, tell me more about the actions. And Marnie is wicked smart. And she's like, tell me more about this. microglia cell. And then we started talking, and it's very evocative. And then she said, follow the story of this cell, make it your main character, and bring it to life. And I think it matters because it allows us to talk about all the things we talked about tonight, which shift a paradigm that's been around for a very long time. And as we shift that paradigm, we begin to ask new layers of questions that will bring new hope for patients. And it's time. Patients are caught in this liminal space of despair. And that's why I think they matter. They're the main character to tell a story that's never been told and that needs to be told for easing suffering. That's what I got.
Does it for me, Beth?
Yeah, well, I just want to thank Donna for deciding to make microglia her main character and to bring recognition and discussion around my very favorite cell at the moment. I'm slightly biased, but for me as a scientist, how is it that this represents 10% of the cells in our brains? And how is it that only over the last decade or even more recently have we only begun to understand the mysteries of this cell? So I'm a scientist because I really want to understand how things work. There is so much to learn, and so that drives me. But on the flip side, just beyond the basic science part of it, to know that something that any of us uncover in the laboratory could lead to a new therapy or a new approach that could open up all these different areas to help patients, that's very motivating, it's driving me to want to work harder and faster to try to figure it out even more than I did if I was just curious. So the curiosity is what drives me, but knowing and hearing the stories of these patients and how it could have a higher, broader impact continues to really motivate me and many other scientists. So we're really grateful also to be able to have this out in the more public venue so people can start to think and talk about it. Thank you.
Well, I have to give it back to her because she let me tell her story. And there are lots of emails that you never see behind the scenes where I'm like, Beth, does this sound right?
Oh, yeah, I know.
And I'm like, no.
No.
That's
and
so
then
wrong.
I go, how about this? And we did a lot of yellow.
A lot of red.
A lot of red and yellow. And, but she's that amazing scientist who doesn't change your paragraph. She changes the words that need to be changed. And it can be otherwise.
And I would very highly recommend this book. It's a combination of beautiful science writing and really vivid and moving human stories.
Thank you.
So books upstairs, food upstairs. Please join me in thanking Donna and Beth.
And thank Carey. Thank you so much for coming out. Thank you.